Good morning, it's December 6th, and this is your daily brief in science. Here's everything you need to know. Billionaire Ken Griffin has made headlines with his recent purchase of a nearly complete Stegosaurus fossil, nicknamed Apex, for a staggering $45 million at auction in July 2024. This acquisition marks a new record, surpassing the previous high of $31.8 million for Stan, a Tyrannosaurus rex sold in 2020. Apex, discovered by commercial paleontologist Jason Cooper in May 2022 near Dinosaur, Colorado, is one of the most complete specimens ever found, with approximately 80% of its 320 bones preserved. Measuring 11 feet tall and 27 feet long, Apex is expected to be a significant attraction for museum visitors. The fossil will be on public display starting December 8, 2024, initially in the museum's atrium before it moves to the fossil halls next year. Griffin has also committed to a long-term loan of Apex to the American Museum of Natural History, which will allow scientists to study the fossil and conduct analyses. Research initiatives will include CT scans of Apex's skull and an examination of its thigh bone, aimed at providing insights into Stegosaurus metabolism and bone growth. According to museum curator Roger Benson, Apex is well preserved due to being buried quickly and not extensively disturbed. This preservation may help researchers understand the species' growth and metabolic rates, as Apex is believed to have died at an old age. In addition to the loan, Griffin has pledged funding for research and educational programming related to the fossil, ensuring that millions of visitors will have access to this important specimen. However, the purchase has sparked concerns within the scientific community about the implications of privately owned specimens on research access, with ongoing discussions about the ethics surrounding such arrangements. A groundbreaking study published in Cell Stem Cell has unveiled that generating new brain cells in adults can significantly enhance verbal learning and memory. This discovery may pave the way for innovative therapeutic options aimed at cognitive restoration. The research, led by scientists from the USC Stem Cell and USC Neurorestoration Center, identified a crucial link between the decline of immature brain cells and the deterioration of cognitive abilities in patients suffering from drug-resistant mesial temporal lobe epilepsy, or MTLE. This marks a novel finding in the field of human neurogenesis. To explore this relationship, researchers analyzed brain tissue from epilepsy patients, focusing on how adult neurogenesis correlates with cognitive performance. Current treatments for epilepsy primarily aim at controlling seizures, often neglecting the associated cognitive decline. In response, the team has initiated a clinical trial to investigate the potential effects of aerobic exercise on neuron production and cognitive function in epilepsy patients. This trial is currently in its early phase 1 stage and has reported initial positive results from 2 participants. Co-corresponding authors Michael Bonaguidi and Charles Liu stress the importance of neurogenesis in cognitive functions, particularly for individuals with epilepsy. They highlight how new neurons contribute to learning, emphasizing the need to understand their function in humans to support brain health throughout life. The study also underscores the necessity for human research on neurogenesis to validate findings from animal models and ensure the clinical relevance of treatments aimed at cognitive enhancement. Utilizing surgical specimens donated by patients from the Rancho Los Amigos Epilepsy Center, this research also contributes to health equity among underinsured populations. Funding for this significant study has come from multiple sources, including substantial support from the National Institutes of Health, showcasing a collaborative effort to advance health equity and research. Overall, these findings open new avenues for future research aimed at improving cognitive function through neurogenesis, potentially benefiting a wide range of individuals facing cognitive impairments. In Sweden, approximately 9,000 women are diagnosed with breast cancer each year, with hormone-sensitive breast cancer accounting for around 75% of these cases. A recent study led by Linda Lindström from Karolinska Institutet has revealed that postmenopausal women with low-risk estrogen-sensitive breast cancer can benefit from anti-hormonal therapy for over 20 years. This stands in contrast to younger, premenopausal women who tend to experience only short-term benefits from the same treatments. The study analyzed data from over 1,200 women diagnosed with hormone-dependent breast cancer between 1976 and 1997, including nearly 400 premenopausal women. Findings indicated that women with high-risk tumors had less benefit from treatment, regardless of their menopausal status, while those with low-risk tumors in postmenopausal women showed significant long-term advantages. Notably, younger women, particularly premenopausal patients, face a higher risk of recurrence compared to their postmenopausal counterparts, yet most studies have historically focused on the latter group. The researchers emphasized the urgent need for new markers to predict treatment benefits and assess long-term recurrence risks in younger patients, as existing markers have proven ineffective. Future research aims to individualize treatment strategies for younger patients, enhancing their long-term benefits from anti-hormonal therapy. Upcoming studies will employ advanced techniques such as multi-protein analyses and machine learning to better understand tumor characteristics and their impact on treatment outcomes. The study received funding from various Swedish research organizations, and researchers reported no conflicts of interest. Participants were randomly assigned to receive tamoxifen for at least 2 years, or to a control group that did not receive anti-hormonal treatment, allowing for a unique comparison of long-term outcomes. While anti-hormonal treatments like tamoxifen are effective in suppressing estrogen-driven tumor growth, they can negatively impact quality of life, underscoring the need for ongoing assessment of long-term benefits. A recent study has uncovered a notable connection between the genetic risk of depression and cardiovascular disease, particularly impacting women. This finding is significant, especially considering that men generally have a higher overall incidence of heart disease. The research analyzed genetic and health data from over 300,000 individuals, revealing that the risk of coronary artery disease persists even in women who have never been clinically diagnosed with depression. The study, published in the journal Circulation, Genomic and Precision Medicine, consistently identified cardiovascular risks in women, irrespective of their menopausal status at the time of recruitment. Conducted by Dr. Sonia Shah and Dr. Clara Jiang, the research emphasizes the critical differences in cardiovascular risk between genders. Dr. Jiang noted that women have historically been underrepresented in cardiovascular research, which has contributed to biases in diagnosis and treatment of heart disease in this population. Heart disease remains the leading cause of death globally, with 1 in 3 deaths attributed to it among Australian women. Furthermore, women with heart disease are often understudied and undertreated, leading to higher mortality rates from heart attacks compared to their male counterparts. Dr. Shah highlighted that the link between depression and cardiovascular risk is evident even in women who have never been diagnosed with depression or used psychiatric medications. Currently, only the QRISK-3 calculator in the UK incorporates depression into heart disease risk assessments, but it does not differentiate the impact of depression between sexes. This research underscores the urgent need for a continued focus on understanding heart disease in women, advocating for tailored approaches for prevention and treatment. A groundbreaking discovery has emerged from Lake Enigma in northern Victoria Land, revealing a significant body of unfrozen water beneath its ice cover. This finding challenges previous assumptions that the lake was entirely frozen. Researchers from the Italian National Research Council utilized ground-penetrating radar, which indicated that the water lies at least 12 meters deep, with some areas potentially even deeper. The radar survey uncovered a complex hydrological system beneath the ice, suggesting that Lake Enigma contains areas of unfrozen water exceeding earlier depth estimates. Despite its location in one of the driest regions on Earth, the lake experiences limited ice melt during the summer, losing between 70,000 and 200,000 cubic meters of ice annually due to sublimation and evaporation driven by katabatic winds. Lake Enigma, formed during a warmer climatic period, has average annual air temperatures around -14 degrees Celsius, with winter lows dropping to -40.7 degrees Celsius. The sediments in the lake are primarily composed of silicate-rich minerals, indicating ongoing chemical weathering processes under isolated conditions. Notably, microbial mats found at the bottom of the lake exhibit diverse structures, suggesting a stable environment that supports a unique microbial ecosystem. Genetic analysis of the microbiota revealed a variety of microorganisms, including Pseudomonadota, Actinobacteriota, and a notable presence of ultra-small bacteria from the superphylum Patacibacteria. Researchers propose that these microbial species may be descendants of an ancient ecosystem that existed before the lakes freeze up, indicating a simple aquatic food web. The study aims to enhance understanding of Antarctic biology by comparing Lake Enigma's geochemistry and microbiology with other lakes in the McMurdo Dry Valleys. The findings of this research were published in the journal Communications Earth and Environment, shedding light on the ongoing mysteries surrounding Lake Enigma and suggesting the existence of an unknown water source, possibly a hidden recharge system. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.